So I'm here to transition us into animal life. What we've seen is that there has been a dramatic transformation of the human brain just in the last 3 million years. And let's go back to see what led up to that and then start to speculate where we might be going. We have some big decisions ahead of us. In particular, already cities and the internet are kind of transforming the way our brains work. What's ahead? So let's take a very conservative 3.8 billion years for DNA-based life. We have an enormous variety of species on this planet. Let me make a pitch. We should be sequencing them. It's a crime to let these species disappear without at least reading their DNA. It's a crime to let them disappear at all. There are a number of projects to do that. So the tree that Carl Rose set out gives us our 3 branches. And of course, the mysterious thing is that eukaryotes seem to be a combination. And this was, of course, Lynn Margulies' beautiful hypotheses, which she submitted a dozen times for publication before it was actually published in 1967, that in fact an archaeobacteria and or a archaea and a bacteria combined to make our modern eukaryotic cells. Now, these have incredible machines, and you heard from Karen and from Gary, some of the most marvelous early machines. The eukaryotes just went crazy and invented all kinds of more motors and complexity that we don't have time to go in. Probably their most fundamental inventions were sex and death. So this is something that is a profound transformation if we think about it in terms of how that affected life. Is this a necessary thing if we look for life on other planets? At the time the eukaryotic species were being formed, we were just coming off the great oxygenation event. And we have the Huronian glaciation right after that, probably timed very coincidentally, interestingly, with the eukaryotes. Right after that, the boring billion. That's always amazed me, right after the invention of sex. So what happens? What does it take to get to the next level in evolution, and what happens? Probably there were many, many more machines being invented that left no fossil record. We see 800 million years ago, the common ancestor of the metazoa, the multicellular organisms, in definitive form, sponge-like form. And now we're already billions and billions of years after the times we've been talking about for most of this talk. And then, bam, another glaciation. The Sturtian glaciation. And again, that corresponds with an opening of life. First, the Ediacaran, and then the Cambrian Explosion. So there's some kind of radical climate change. Of course, the great events often caused by life themselves that lead to these spectacular profusions of new complexity and form, and predation. So one of the other things we think about when we look at communicating and interacting with life on other planets is this rich complexity inevitably associated with predation. It seems to appear at this time. So I'm going to give you a quick tour of our ancestors. This is who's your granddaddy, all right, starting at the Cambrian period and then going on, Silurian, Devonian, Permian, Cretaceous. Now we're 70 million years ago, and we've seen a colonization of all of the ecosystems in the planet and in the oceans. And now, if you think about this, the rise of the placental mammals after the great catastrophe slightly before this at the KT boundary, creates this tree that we're now looking at here. And you can go to the UCSC genome browser to see most of these genomes. We're thinking about genomes in action, but I'm just showing you pictures of living individuals at this point, all descendants from the common shrew-like ancestor 70 million years ago. If we go through them rather quickly, you can see as we march forward along this tree to branch points, you can see the evolution of beautiful forms, animal forms that now become more and more human-like as we are going towards our human form. What were the molecular transformations that took us from one species to the next here through this time period, finally reaching human? So all of these changes were caused by DNA mutations, all right? The amazing story of this is it's all edits in the script of life that controls our cells. We can reconstruct what happened at various times. We can even turn this process around and de-extinct species that have gone extinct. Here's the demise of the gene as seen from the UCSC browser. All it takes is a one base change to create a stop codon, and bam, that gene is gone. And gone forever unless we engineer it to come back. So with that as an introduction, I want to focus on the main part of this. Humans have a much larger brain, so 3 times as big as it was 3 million years ago. How did this happen? Well, we know that there are regions of the genome that have been associated with brain disorders that harbor genes that are very important for the development of our gene, and we get diseases when we don't have the genes working correctly in these regions. Microcephaly, too small a brain, macrocephaly, too large a brain, are logical consequences of misfunctions in the genes that may have been associated with our larger brain. When we were actually comparing early neurodevelopment in the rhesus to humans, we noticed this gene, Notch2L, is highly active in the humans. And I remember when my postdoc, Frank, came to me and said, yeah, but it isn't even in the rhesus monkey. I thought, wow, this is incredible. Here's a gene that's very recently evolved. Well, it turns out that only humans have a functional form of this. The reason it wasn't studied, people always ask me, why wasn't this studied? It was in the wrong place in the human genome. My apologies, again, there, right? We were part of the Genome Project. But this is an incredibly hard piece of the genome to sequence. And in fact, we had Notch2L on the other side of the thrombocytopenia absent radius region, which has a very distinctive phenotype, this truncated limb, and not anywhere near the macrocephaly, microcephaly region. But it turned out that was wrong, and in fact, the gene is right there. So not only is it a new human gene, but it has the phenotype we would expect. We went back and sequenced all of this DNA data that had been mapped to the wrong reference genome, and when you map it to the right reference genome, you see that the macrocephalics and the microcephalics have deletions in the case of microcephaly, duplications in macrocephaly at exactly this region. So further digging shows that this gene was actually created in the early on. A piece of it was chipped off of an existing protein, Notch2, but it was only revived to become a functioning gene before the split with human and Neanderthal and Denisovan. So all of these species do have much larger brains. The gene duplicated 2 more times, so we now have 3 copies. If you look at fossilized cranium, this period of initially reviving the gene and duplicate it corresponds exactly with endocranial volume measurements. So bottom line is, this gene is very likely to have played a role in the evolution of our larger cerebral cortexes. This, of course, happened so that we could survive in the wild and not so we could understand quantum mechanics. So what was going on? Why did this happen, and how does it make our brains the way they are? And why do they do other things like quantum mechanics? Still a great mystery. One of the things we can say about this is that one of the key thing that differentiates us from other modest brain-sized species is we have a lot of outer radial glial cells. So the cerebral cortex is a multi-layered structure, same number of layers in our primate cousins, but we have more outer radial glias. And those cells can actually be modeled in the lab with an organoid. And so the last part of my talk, I'm going to tell you about cerebral organoids, which we've been growing now for 8 years in my lab. So these are facsimiles of actual neural tissue as it develops in the early embryo. They create layered structures like the cortex, and you can examine them for the cellular and molecular properties of them. So what we did, of course, was take CRISPR-Cas9 and knock out the gene notch to an L, excuse me, and we saw a dramatic change in this organoid development. Essentially, the organoids were stunted, and they fewer outer radial glial cells, and exactly the phenotype that we would expect. I was incredibly excited here because we had an experimental model now. Here's a process, the determination of cortex size and the early evolution of the cortex that clearly happens at a stage that's very early in development, and now we can model it in the lab. So what are these things? Cerebral organoids. They really are, there's an amazing confluence now of technologies that will allow us to reverse engineer human brain evolution and function. So the genomes and all of the great molecular biology methodologies that have been developed are fundamental. The thing is that cells, embryonic stem cells, have an extraordinary property of self-assembly. So if you give them the right environment to convince them essentially that they should be forming a cerebral cortex, they do. Here's an example for the murine eye cup. If you look at this optic cup, you can see in a dish on the right spontaneous formation of the cup-like structure. So the genome is programmed to develop from a single cell ultimately into an entirely fully structured animal organism. The early cortical layers that we can create ex vivo in a dish, as it were, include all of the key cell types that you find in neural tissue. And if you grow them more than 10 weeks, you start to see electrical activity as well. So we think we can model circuits in these. So I would ask you to think about, well, we're talking about making communication with alien life. Maybe there is some kind of alien intelligence we might interact with. At the same time, we're developing our own AI on this planet. How are we going to understand how the way we think, is different or the same compared to these other types of intelligences? The only way we're going to do that, I claim, is through a platform that is experimental, and I think cerebral organoids are it. So we're currently able to grow the organoids. We have methods for testing them, electrical connections, various arrays and probes. We use Optogenetics, a wonderful methodology, not invented in our lab, but it allows you to actually create, to force a neuron to fire by giving it a light signal. Here's some early experiments on organoids. So the thesis is this. Currently, if you look at neuroscience, the dominant model is behavioral neuroscience. Here you have a mouse that's wired up with an optogenetic rig. They're stimulating neurons in the mouse's cortex, and they're looking at behavioral results. Based on that, they want to build a in silico simulation. So we don't understand it unless we can really model it completely. I claim that that gap between brain activity in a living mouse and an in silico simulation is too large for us to manage in any reasonable amount of time. These experiments are expensive and they don't scale. And further off, it's a mouse. So what we're doing right now is looking at actual human brain sizes, and we have this back and forth loop to model them with organoids. The organoids get better and better and better all the time, and we can check to see whether they're recapitulating activity in actual human brain slices. Excuse my cough. Then, at the same time, we can grow thousands of organoids and make models and actually interact with them computationally and get to the point where we can reproduce their behavior. So these, by inserting organoids in the middle of this, we have a reasonable stepping stone that is fully experimental and human that may get us there. So speculation, all right, so sci-fi moment, right? So we have the graphical processing unit, right? We have the tensor processing unit. These drive our computation now. At some point, we'll have wired organoids. There's no reason when we have thousands of them that they won't become objects on the Internet of Things that you can interact with. So the future really holds a merging of silicon biology in a fundamental way. We will be able to actually understand tissue by interacting with it experimentally and in a profound and scalable way in the future. So I don't know what's going to happen, but we'll find out what would happen if you grow a cerebral organoid and let it interact with an advanced AI. I don't know. What if you grow 100,000 of them? These are the questions that are fascinating at this point. And it follows on Steve's comment about, well, you know, maybe we can invent it here and interact with it here. That's one thing when you're looking at chemistry. It's another thing when you're actually looking at cerebral cortex. So we'll see what goes on with that. Here are the brain engineers at UCSC and our colleagues at UCSF. Thank you.
Questions, even if it takes time. We have to take, it's a discuss. We're a discuss, right? 1 or 2 questions. Thank you.
So if you read the literature, people talk about, I don't know, ecological drivers avoiding predators or cooperative behavior driving brain evolution. Are you saying that this was just a random thing when this notch gene duplication event happened 3 to 4 million years ago, and if it had not had happened, we'd still be scraping around on the savannah, and we wouldn't be doing quantum physics?
Possibly, yes. The other option is if this hadn't happened, some other thing would have happened, but probably not on the same time scale.
So you would dispute these ecological
today,
drivers?
In 2019, we wouldn't be doing quantum mechanics. Yes, probably we would not be doing quantum mechanics in 2019 if this event did not happen 3 million years ago. Okay. Yes, I would definitely say that. But it's not to say that we wouldn't be doing quantum mechanics in 100 or 300 years because of some other event. We were definitely poised to become more intelligent. I'm not saying this is the only way you can.
We're taking one more quick question.
So, is the singularity near?
Yes. So, I think there will be profound effects when we start to interact with AI and interact with our own biology in a way that we understand it much in a fundamentally deeper way than we do now. I don't believe in a pure singularity in the sense that it's more an S-curve with all of these technologies than a point singularity. But we will see profound and accelerated change, yes.